second. All right, so it's my honor and pleasure today to introduce Dr. Vladimir Beljanski, who is opening our spring 2020 Coulter seminar series. Dr. Beljanski is just off the street from us at Southeastern University at the Cell Therapy Institute. A little bit of background of Dr. Beljanski. He completed his PhD at Emory University, and then subsequently he moved on to a postdoc at South Carolina. And he has been at the Cell Therapy Institute for the last 5-6 years, where he's currently a clinical research assistant professor. His area of research expertise it spans a number of cellular areas, but I think most recently, and the focus of his talk today, is on mesenchymal stromal or mesenchymal stem cells, and examining their immunological properties, immunomodulatory properties, as well as I found out just now, looking at their exosomal production, which is also very exciting for us because we're sort of doing similar things. In addition to that, he's involved in a variety of other activities as far as looking at cellular responses for various applications, including cancer and other things like that. And he's also coined the term the Miami cell, but I will not steal his thunder there, and I'll let him explain. I'm sure Miami cell is part of the course here. So please join me in welcoming Dr. Beljanski.
So the first thing I'd like, everybody can hear me, right? Because I'm pretty loud, so people always tell me, speak softer, you're loud. Anyway, so thank you, Dr. Ramaswamy, for the invitation and for letting me present my research today, and thank you to the Department of Biomedical Engineering for also making all this possible, and thank you all for coming to hear about a research project we're doing at NSU. So NSU, I don't know how much you know about it, it's located about an hour drive from here in Davie. It's not really it wasn't really a research institution. They started to dip into research in 2012, and they were subsequently bringing researchers from different parts of Florida. This is how I ended up there in 2015 from an institute that was located in Port St. Lucie, just north of West Palm Beach. So we opened our labs in 2017, and the project or the data that I'm going to present you today is my first senior author publication from the work we studied sometime in the summer of 2017. So the talk is really about two different things. And when I started working in this area, which was about 2015, 16, 17, my first job was really to develop therapies for cardiac regeneration. Now I come from the field of cancer, so we study cell death, cell survival in cancer, and I knew nothing about cardiac regeneration. So my first thought was, how can I implement my knowledge of cell survival, cell death into the field of stem cell therapies? And then I started looking at the literature, and I realized no one had ever looked at how cellular cell survival, cell death impacts on the efficacy of stem cells. So this is what my talk is going to be about. This is what the seminar is going to be about. So as I said, two things. One is we are currently beginning more and more to use stem cell in tissue regeneration and therapy. But we haven't really looked at what really drives therapeutic efficacy of these approaches. And as I said, given that I come from the field of cancer, I was interested in this pathway, and I don't know how much you have studied, how much you have heard about it, called autophagy, or cellular self-eating. And autophagy is the easiest way for me to explain is when you want to lose some weight and you go on a diet, you're really undergoing autophagy as an organism. Well, individual cells are doing exactly the same thing. They're digesting themselves in these vesicles, yellow, reddish vesicles that you're looking at here, is really cell undergoing self-starvation. And if you think that this is not that important, just a reminder, 40 years ago, Japanese scientist Dr. Oshumi received a Nobel Prize for dissecting the mechanism of autophagy. So what is a stem cell? So let's start, and I know this is bioengineering, and I probably shouldn't be talking about it, but this is kind of like a standard way I like to look at that. So what is a stem cell? Any cell that is in sort of like a more primitive, non-functional state that can either replicate or differentiate is considered to be a stem cell. And I love this slide because it really talks about how general public perceives stem cells, right? So they think that stem cells is this really amoral work in which you have this cell that if you leave it in a petri dish, it's going to give birth to a living organism or a living baby. And I got this cartoon, I don't remember, like years back, and it's not true. And I always tell, whenever I go to talk to the community of lay people, I tell them, you all have stem cells. We all have stem cells in our bodies. What is really ethically questionable is embryonic stem cells. But other stem cells that we all have, bone marrow stem cells, adipose-derived stem cells, are not questionable when it comes to research and doing therapies with them. So that being said, if you go to clinicaltrials.gov, you're going to find that there's quite a few of clinical trials happening all over the world. So this is all registered clinical trials, and when you go to the search term stem cell, this is the number that you get. Given what the promise of these therapies, this is a low number. So we are working toward doubling or maybe tripling these numbers. We want to show that there is a lot of things that you can do with stem cells. So what is known and what's been used for many decades is hematopoietic stem cell transplantation. So person who is, let's say, had leukemia, took drugs to ablate their immune system, now is going to have their immune system reconstituted by giving allogeneic bone marrow transplant from a healthy individual, and hopefully this person is going to develop this new individual's immune system. And that's shown here. So this is a patient. This is where the cells are typically stored in liquid nitrogen water bath, and basically he's injected with a new hematopoietic stem cells to reconstitute his immune system. Well, that's been standardly used in clinics. So what is the promise of this? Promise is enormous. A number of diseases that can be treated with this approach is huge, but this is the promise, and between the idea and the execution lies a shadow. So that shadow is huge here, and we have to work on overcoming that and really using appropriately stem cells to treat, if not all, but then the majority of these diseases. So let's start with mesenchymal cells and talk about what are they and for what they can be used. We all have them, right? So at some point, we were these three layers as we were developing from one cell into what we are now, living people. So it is believed that not all cells from mesodermal layer have developed completely. They have not all differentiated. And the stem cells, the mesenchymal stromal stem cells that we have, are those that have not properly differentiated. But we have them in almost every organ. But we're interested in them because they have therapeutic properties. Their role in our bodies and our tissues is to sense inflammation and shut it down. This is what they can do. Well, the problem is that except for your bone marrow and your adipose tissue and umbilical cord, for those who have given birth, don't really have much of these cells. So what you have to do is you have to add them exogenously. So you can isolate from multiple organs, as I said, bone marrow, adipose tissue, peripheral blood, placental tissues. Derived MSCs are fantastic, but how many placentas can you find? So most of the cells that are used right now in therapies are isolated from bone marrow because you can get a lot of them. There's no ethical problem here. You can easily expand, you can maybe differentiate, and then inject it to people. There are problems with it as I'm going to address, but this is being tested as we speak. This procedure has been tested. So as I said, what do these cells do? Their job is to sense inflammation. When they sense inflammation, they become usually, not always, but usually anti-inflammatory, which means they're shutting down T-cell response. So they're moving, they're changing helper T-cells into regulatory T-cells, and by doing that, they're shutting down inflammatory response. An inflammatory response is frequently seen with tissue damage. So this is how they work. They don't magically go and differentiate the tissue that is missing. What they're doing, they're helping endogenous cells become better by secreting factors, by shutting down T cell response. That's what they do. And they do it really well. You just don't have enough of them. This is how they do it. Not only did they secrete exosomes and proteins that are shutting down T cell response, they're also on their surface expressing PDL, programmed death receptor ligand 1, and that one is going to really shut down T cell response. This is the very first, it was published in Lancet in 2004, clinical application of MSCs that was shown that can be efficient and good for a patient. This is Katrina LeBlanc from Karolinska Institute, and she's a pioneer in using these cells for therapies. She's an hematologic oncologist. She had a pediatric patient who was on a second remission, and he had hematopoietic stem cell transplantation. He developed graft versus host disease of the gut and the liver, and was not responding to steroids and cyclophosphamide. So they did compassionate treatment. She was studying MSCs to shut down inflammatory response, systemic inflammatory response. They injected the patient twice with MSCs that were donated by his mother, and boom, the inflammation was gone. When I saw her 3-4 years ago, I asked her, so what happened with that patient? The patient died of leukemia, not of the graft versus host disease, because he was in the second remission, and then after this therapy, he lived for another 2 years, and then they redeveloped leukemia, and they couldn't save him. So overall, the story is really sad, but this has shown to us that there will be people, there will be children who can be successfully treated with MSCs, because the treatment here was really to offset this graft versus host disease that almost killed him. So as with any therapy that is being developed as we speak, there are issues. There is a quality of tissue regeneration issue. Cells that are injected will not survive for a long time. They are experiencing adverse environment when they're injected. So I'm talking about MSCs being injected into a patient. They are not being welcomed by our organism. So they will not survive for a long time, and they will definitely not engraft. There is immune response against them. Not a strong one, but there can be an immune response. Should we use autologous allogeneic stem cells? We're testing it. We don't know yet for many applications. They're enormously expensive, these therapies. Effectiveness goes to the number one, the quality of regeneration, and then finally safety. Safety so far has not had any, has not given us any problems, but we don't know that yet. I mean, we're still in the phase of looking at that. Now let's switch gear. So now we found out that there is this potentially very good therapy that has a lot of issues, right? So remember, we're talking about living cells being injected into a living organism. These are not small molecules. So what is this got to do with autophagy? Well, autophagy is a process that helps cells survive when they're exposed to adverse conditions, be it advanced immune response, be it lack of stromal support and lack of nutrients. And if you ever think about reading about autophagy, I'm going to give you a preview that those who are activating their autophagy, I'm talking about people who are activating their autophagy frequently by not eating too much, will live longer. Now let me ask you something. What is common for all people that live longer than 80 and 90 years? What is one thing they all have in common? Doesn't matter where they are, what nation, what background. What is this one thing they have in common? They're all skinny. Exactly. There is a reason for that, why they're living so long. Because what autophagy does is it helps cell regenerate itself. It basically regenerates organisms, improperly folded proteins, damaged proteins, better than any other pathway that we have. So this is a great scheme that I stole from Dan Klionski's review paper published several years ago in 2010. Well, now it's a decade long. where he's showing that where autophagy is going to have role in the cell, right? And if you look at these five processes that autophagy can impact on: metabolism, immune response, fighting oxidative stress, increased cellular remodeling, and fighting starvation. These are 5 things that are important for injected stem cell. So their oxidative stress goes up, they're maybe encountering immune response against them, they're undergoing starvation, they don't have strong support. So my idea here was, and it brings us to the, well, not yet to the hypothesis, but I'm going to tell you a little more about molecular autophagy. But my idea was that no one ever looked at the role of autophagy in these cells. How can you increase therapeutic efficacy? Can you increase therapeutic efficacy by manipulating how these cells handle their stress? So let's get into a little more about molecular players in autophagy. So 3 most important players in energy sensing: AMP kinase, AKT, and MAP kinase. So when there is enough of energy for the cell, they're all happy. So it's a high energy, they're activating mTOR. mTOR is blocking ULK1 kinase, it's blocking this autophagy initiating complex, and everything is going fine. Proteins are synthesized, cells are happy. However, during starvation, when there's not a lot of energy, AMPK, which senses ratio between ATP and AMP and ATP, is going now to block mTOR, avoid protein synthesis, but it's going to block this ULK kinase, and it's also going to activate protein complex that is going to create triphosphate autophosphatidylinositol. And then when that happens, this small molecule, and this usually happens very close to the ER membrane, this small molecule is going to change the shape of the ER membrane, and by changing this shape, creating these so-called omegasomes, it's actually going to start creating this phagophore, which is an isolation membrane that now is growing out of ER membrane, and it's going to incorporate this light chain protein 3, microtubule-associated light chain protein 3, into the autophagosome, and I put it purposely in green because we usually use this as a marker of autophagy. So you can stain it, or you can tag it with GFP and find where the autophagosomes are. Now, when this phagophore is formed, what it's going to do, it's going to start engulfing cytoplasmic material with other proteins that belong to the autophagy pathway. It's going to grow and elongate, and then finally it's going to create autophagosome. Okay, so this is now a little vesicle inside the cell that contains proteins and sometimes whole organelles, sometimes mitochondria. And that is going to now fuse with the lysosome, and with the lysosomal hydroxylases, it's going to break down these proteins, creating basically small molecules that are going to be fed into the metabolism. So this process is going on and on and on until there is enough exogenous glucose and nutrients for cell to stop and rely on exogenously added nutrients. So the good thing about autophagy is that it's druggable. And I've shown some of the drugs that you can use. A lot of drugs that cause autophagy were not developed to cause autophagy. It just turns out that they're causing autophagy or blocking autophagy. For example, chloroquine is an antimalarial drug. It's been known forever. What it does is it acidifies lysosome and it blocks fusion between autophagosome and lysosome. There are some kinase inhibitors that are blocking the very first kinase complex that creates phosphatidylinositol triphosphate. There's metformin that actually activates activity on NPK, and some others. A very known drug in autophagy is rapamycin, because rapamycin is blocking mTOR. The bad thing about autophagy is it's reversible, so you kind of have to have drug there all the time. So now if you think about cell that's been isolated, processed, and then finally injected into patient, and if you think about what is going to happen when you just do what's shown here, or when you actually activate or block autophagy, what's going to happen is if for example, it's just injected cell, it's going to be detached from the matrix because it doesn't really belong to that organism, so they're going to go apoptosis. It's going to probably secrete some of the cytokines due to the high levels of ROS. It's going to impact on CD4 T cell, it's going to recruit immune cells, and that's going to happen without modulation of autophagy. However, if you modulate autophagy, you may actually help cellular remodeling, number one. Number two, you're going to decrease ROS, you're going to decrease secretion of cytokines, and thus you're going to decrease immune response against these cells. So that led us to the hypothesis that if you modulate autophagy, you can actually change the way that these cells are responding to inflammation, and thus change the way that they are going to be, change exactly their therapeutical effects or their therapeutical efficacy. So what we did, we used these Miami cells, and I have to say it wasn't me who coined the term Miami cells. They were isolated by Dr. Paul Schiller from Miami VA, and I just happened to put a project together with him, and that's how I found out about these cells. They are a subset of MSCs. So MSC is a collection of different kinds of cells. One of those cells that are found in MSCs, in bone marrow MSCs, are called Miami cells. Marrow isolated adult multilineage inducible cells. The difference between those cells and MSCs is these you can grow for a long time and you can pass them for a long time, as opposed to MSCs, which is going to start dying after the passage 10 or 12. Anyways, so this is the characterization of these cells. They've been isolated in 2000, they've been published in 2004. And what we did is two things. First of all, we do a lot of systems biology, so I was interested in gene expression. And we also do some functional assays. So the gene expression, what we did is we took the Miami cells, we exposed them to interferon. That's our, stimulation of inflammation in a petri dish. Then we modulated autophagy with drugs. And then did the RNA sequencing, all that good stuff, then used IPA analysis, and found out how addition of autophagy modulators change, and whether it changes, the way that these cells respond to inflammation stimulation. Okay? So we used two drugs. We used tamoxifen and chloroquine. So tamoxifen, its mechanism is a little more complicated. Chloroquine, we know. It just acidifies lysosome, and good, lysosome is incapacitated. But tamoxifen has a little more complicated mechanism of action. I put it there as an MPK blocker, but there's some evidence that tamoxifen really elevates ceramide, and that's really how it causes both apoptosis and autophagy. So we expose cells to the following treatments, so either interferon alone or together with tamoxifen and chloroquine on day 0, then for 4 days collected RNA and performed RNA sequencing. Okay? So this is just to show that we actually didn't have cell death in these treatments. We had activation of autophagy. This is blocked. This is looking at the LC3 cleavage. Whenever you have this bottom band going up, that means that there's increased level of autophagosomes. We also measured this with cyto-ID, shown here. So this is cyto-ID data with flow cytometry, confirmed that. And then our gene expression analysis. So what we are finding out, so the very first bar here, the white bar, right here, the white bar, that's just cells exposed to interferon. However, the bottom bars are chloroquine and tamoxifen. So what we're finding out is that if you add autophagy stimulated tamoxifen, autophagy blocker, chloroquine, you're changing the gene expression. You don't necessarily change which gene are differentially regulated, but you change to the extent that they're differentially regulated. So the pathways kind of remain the same. This is the statistical significance of the pathway, which takes into account number of differentially regulated genes and the extent to which they're differentially regulated. What we found out is that we're changing the way that pathways contribute to the response. But we don't change the pathways. We change the way the genes are going up and down, but we don't change the genes that are going up and down, except for maybe a few genes here and there. And what's most important here is this. This is a putative upstream regulator analysis, which you can do. I don't know if you guys are using Ingenuity pathway analysis for analysis of gene expression, but this tells you whether different treatments are, trying to explain this the best way. So upstream regulators is a theoretical up or down regulation of genes and pathways that are connected to your data set. Meaning that if you treat with interferon gamma here, these are potentially activators or inhibitor genes that are not necessarily changing your data set, but that could cause the change that you're seeing in your data set, right? Now, when we add chloroquine and tamoxifen, we're not seeing a big difference, which tells you that the nature of response remains the same, even though it remains the same when it comes to stimulation and inflammation. Even though you are changing some of the gene expression, you're changing the contribution of the pathway, the nature of the response of these stem cells to inflammation remains the same. And that's what we wanted to see. We want to modulate this therapy. We don't want to change. We don't want to shut off response to ourselves. So we're very happy to see that. This is some of the pathway network analysis, and I put it here. It's a really nice picture. Reviewer asked us to do it. It's a really nice picture. I don't really tells you much, except that you can see that the way that these pathways are interconnected, and these numbers here, the number of genes that belong to the same pathways that are differentially regulated in your data set. You can see that the way that these pathways kind of interact theoretically changed a little bit, which again, we like to see that because otherwise it would mean nothing really changes, right? But you can take it as a nice picture. We also looked at, there is this great tool that, again, I've learned because reviewer asked to do this, called Web Gestalt, and it's free, and you can incorporate your data set, and then you can look at, not pathways, but you look at enrichment. And enrichment is the number of genes that are kind of doing similar things. And when we compared, this is biological process, cellular component, molecular function, and when we looked at the difference between the two treatments, there is none. And we like to see that. You're not changing the way the cells are responding to inflammation, you're just changing the extent how they respond to inflammation, okay? We also looked at, because we had a kit to study microRNA, and I'm not really a microRNA expert, but my partner who did all the RNA sequencing, she has these fun, fancy kits, and also my good friend has made a career in microRNA, so I called her on the phone, and hey, I have this, what should I look at? So they both helped me. Basically, what we found out is that when you measure, the kit measures 800 most commonly used or looked at microRNAs. So we're looking at how these microRNAs are connected to our data set. So these genes here it's an mRNA level for those genes that are participating in immunoregulatory properties of stem cells. Remember I was telling you PD-L1 that express cytokines and enzyme. These are the ones, okay? These are antigen presentation genes, HLA. So I haven't shown every one of them because there is, I think, 30 of them. But the point here is that when you co-treat these cells with tamoxifen or chloroquine, you're changing microRNA that are affecting gene levels. So part of the reason, why we're seeing these differences is because these drugs will also change the expression of microRNAs, and that affects gene expression. All right, so this is just confirmation. I don't know if you guys have NanoString, probably you do. NanoString is a really cool method. You don't really have to spend weeks doing qPCR if you want to confirm your sequencing analysis. You can just put everything one day. NanoString costs you a fortune, but you get your data very fast, and within a couple of days I had this figure generated. Basically, this is confirmation of the RNA sequencing data, and when a person who did Lubo, when she did RNA sequencing, she told me, look, I use 4 aligners, and I only put those genes that are common in at least 2 aligners. And when I do that, they're all perfectly reproducible, and that's what we saw. So our gene expression data were, I won't say perfectly, it sound cocky, but reproducible to a large extent. Let's put it that way. All right, so as much as we love gene expression, it's only telling you one side of the story. Without some kind of functional assay and whether you can prove that your autophagy modulation changes the functional level, you have nothing. So we did functional assays, and postdoc Fiorella, who's the first author on this paper, she's an immunologist. And I purposely hired her in order to develop these kind of assays. So what we did is a classical assay showing that stem cells have immunoregulatory properties. You have a T cell, you activate it with antibody beads, And then you do that in the presence of stem cells, and then you look for proliferation, for T cell proliferation, and as well as activation markers. So in our case, we're not just using cells, we're using cells with stimulated or blocked autophagy. And this is how we did it. So again, our 6-well plate, Fiora likes 6-well plates, so that's why we have these 6-well plates. We seed cells, Miami cells, we have T cells. On the day 3, we're looking for activation marker, and on day 4, we look at the T cell proliferation using flow cytometry. And these treatments, not only that they don't kill stem cells or Miami cells, they also don't kill T cells. So that's important when response we're looking is not because of the cell death. It's actually really tricky to do these things with cells. They are really finicky. So we managed to do it, but I think we're lucky more than anything else. So what we found out is that when we looked at the CD25 T cell activation marker, so you can assess immune cell activation by looking at the expression of their markers. So CD25 is one of those. We found out that tamoxifen really is decreasing expression of CD25, not when we looked at the T cell proliferation, we found out that tamoxifen and chloroquine both decrease T cell proliferation. So you're comparing just T cells with Miami cells or T cells with added tamoxifen on chloroquine. It's not a huge effect, but there is an effect. Now, when we took the media from these experiments, from let's say you take stem cells, you collect the media, so now you have exosomes, you have proteins, you have everything there, and mix that with activated T cells, you get a very good effect on chloroquine. So now remember, there is no stem cells here. This is just the effect of secreted factors. We see even better effect. And then we also looked at the cytokines. So PD-L1 is that receptor that expressed on stem cells that is deactivating T cell. So you can also measure that with antibodies and flow cytometry. We're seeing that especially addition of tamoxifen is increasing PD-L1 levels. We're also seeing this is IDO. IDO is an enzyme that is secreted by stem cells to digest tryptophan. And tryptophan is utilized by T cells for proliferation. So now we're kind of looking at the molecular levels that are molecular factors that are responsible for that observed decrease in T cell proliferation and activation. And so IL-6, we really didn't see much of an effect. IL-6, actually we saw an effect, but it was not statistically significant. IL-6 is one of those cytokines that is secreted by stem cells to shut down immune response. Maybe if we have another, excuse me, if we had another 6 samples, maybe we'll see statistical significance. But right now it's a trend. That's what you like to say this to your reviewers. We didn't see statistical significance, but we saw a trend. So that's good. We saw a trend. All right. So what are the conclusions of some of these experiments? So we found out that IFN-induced changes gene expression can be further modulated with autophagy modulators, chloroquine and tamoxifen. When we performed functional assays, we found that both chloroquine and tamoxifen will help further decrease T cell proliferation and will decrease further T cell activation markers. We are arguing that you can use autophagy modulation potentially in clinics. Now this is kind of like I'd like to say pioneering work because kind of sounds cocky, but it is really novel thing to use stem cell in combination with small molecules. But we argue that potentially you could use this in clinics, maybe not these autophagy, maybe some others, but you could use this in clinics. And of course, the red sentence is always a party pooper. It says, we have to confirm this drug combination in a mouse model first before we do anything in humans. So this is gene expression, this is cell assays, but until you have this proven in a mouse model, you can't persuade anybody that this may work in clinics. And that's what we're putting protocols and shifting into mouse models. So who are people who did the work? So Fiorella is a postdoc. She did the T cell assays. Hunter was our man for everything. He was a lab manager. He has a different job now, but he helped Tirella, he helped me, he did pretty much everything in the lab. My partner, Luba Nathanson, she's also assistant professor at NSU. She's a genomicist, and her technician, Leonor Saria, who did that nanostring validation of gene expression that I show you. Emily, Melissa, and Kim. So these two used to be in genomics facility. They're gone to, this one is now in Mayo Clinic. Marissa Brooks is in the UM, and Kim is helped us with flow cytometry. She's still with NSU. And then I'd like to finish this with 2 slides. So I teach medical students. I'm a professor in the medical school there. And one of my lectures is on stem cells. And I like to show them these slides, which I got by Googling cell therapy clinic and just focusing on South Florida. There is a lot of cell therapy clinics here, not surprisingly so, but I know most of you are too young to think about that, but as you're getting older and older, you think more and more about, should I inject myself with stem cells? I'm gonna live longer, I'm gonna live happier, and a life of higher quality. And to answer this question is this review paper from 2017, still active, from New England Journal of Medicine, that says that the only real procedure with stem cells that is approved and that was shown to have some effect is hematopoietic stem cell transplantation. But other notion and other claims that a stem cell can be injected to treat ALS or multiple sclerosis or anything else really doesn't have a support in current clinical and preclinical practice. Doesn't mean we cannot do it, it's just we don't have data from large clinical trials supporting this claim. And then, another thing I also wanted to tell you is that lay people often ask me, so if they inject a stem cell into my something, will they become tissue of that something? And the answer is no. What we know about stem cells today is that what they do is what I'm showing you that they do. They respond to inflammation and they shut down T-cell response. They don't magically go into an organ and become missing tissue from that organ. There is no clinical evidence. Doesn't mean that it cannot happen, but there is no clinical evidence that something like that can happen. So I always tell when I go to talk to lay people about this, I always tell them, bless you, I always tell them, beware, they're going to take a lot of your money. It's not covered by insurance. There's a good reason why it's not covered by insurance companies. And with that, I'm going to finish, and I'll take your questions.
Thank you, Dr. Beljanski. Questions?
Sorry. So it's really, really cool work. Thanks for sharing.
Thank you.
So I was wondering, at the beginning you were talking about the direct interaction between the stem cells and the T cells versus, I know there's a module out there, you mentioned the exosomes as well. So some people are suggesting that maybe the exosomes are good enough, maybe the synchrotome is good enough. What are your thoughts now? Do you think we need actual cells being injected?
So there's a lot of literature evidence that you can recapitulate quite a few effects of stem cell by using just exosomes. When you're using just exosomes, you're going to miss IDO, you're going to miss PDL1, but you're going to have a lot of microRNAs, you're going to have a lot of long non-coding RNAs that are shown to also shut down inflammation. So I think if you use enough of exosomes, you can recapitulate the effects of stem cells. I think that's how I see that. Yeah. But we don't, like, this is relatively new. Exosomes are even more new, so to say, more novel.
For your mouse studies, they're going to be different, obviously, than your in vitro studies. But do you what do you expect to happen in your mouse studies? I know you expect the same kind of trend, but do you expect?
I'm hoping for the same trend. I don't know if that's going to happen. Mouse studies, I think this was relatively easy to do, not really, but easy to do. Mouse data interpretation of mouse experiments is always more complicated. Are we going to add, so if, let's say we stick with chloroquine and tamoxifen, are we going to add at the same time the cells? Are we going to pre-treat mouse? Are we going to pre-treat cells and then inject them? I'm going to argue it's probably we're going to inject them at the same time. Whereas, for example, this was done in petri dish, so I can control how drugs and cells interact. In mice, most likely I'm going to inject MSCs or their exosomes, inject them through the tail vein, tamoxifen is given interperitoneally, they're probably going to go to the different parts after that injection. So there is a lot of things that I have to figure out before I move into the mouse model to try to, in the mouse model, recapitulate what I'm seeing here the best way possible. In terms of interpretation of the data, chloroquine, for example, has been shown to be immunomodulator on its own. So usually in cancer drugs, because I've done those, this is new for me, but in cancer drugs, what you do is you add subtherapeutic dose. And then you're looking for combination, whether it has some kind of therapeutic effect. In this case, it's going to be trickier to do that. To answer your question, we're going to try a few things, and it's going to be a lot of optimization, a few things that might work. My favorite is really pre-treating the cells before injection, rather than injecting them together at the same time. And if it comes to using this in clinics, and you're thinking about some kind of FDA approval for, let's say, potential clinical studies, they would like that better than injecting them together. Because if you inject them together, you also have to assess toxicity. If you don't inject them together, you pre-treat cells, there might be a way to bypass the toxicity study on its own. So you can bump to the second or third phase of clinical trials without doing the phase 1 and phase 2s.
I have a question,
Yeah.
actually two questions, if I may.
Maybe
First,
three.
the first has to do with your immunomodulation study. You talked about the T-cell suppression. What about perhaps some regulation of both hostile and beneficial macrophage response? And the second question is, do you have any evidence to suggest that these trophic factors that the stem cells release can promote native cell infiltration into the space to promote tissue, since you said the stem cells themselves are not responsible for that?
Well, they're responsible to a certain extent, not too much. So in terms of the first question, in terms of macrophages, I know much less about macrophages than T cells, and when you go to the literature, most people have used T cells, and there's a good reason for that. T cells are really easy to assess, right? So it's easy experiment to do. I know much less about macrophages. I'll probably have to either collaborate with somebody or hire a postdoc who has done work with macrophages. In terms of, tell me again, the second question is the tissue, is there evidence that there is a tissue infiltration?
So, for the notion of using stem cells to mediate de novo tissue production by recruiting native
okay,
cells.
got it.
Through the secretion of factors.
Is there an effect?
Have you seen any?
No, because we haven't done any real tissue experiments.
Okay.
So this was all done in a clean ex vivo.
But even
kind
in
of
the
fashion.
context of factors that have been released, are there factors that have been known to?
IDO. Yes, IDO is known to actually affect that. I didn't really talk about it because it's really in an infancy, but we have an NIH grant where we study the effect of autophagy on exosomes. So we are looking at whether modulation of autophagy in bone marrow MSCs changes content of exosomes. And that's going to be a great, I will know next time I come here with that project, I will know the answer to that question.
Other questions? Well, I'd like to thank our speaker once again for giving us some very deep science in cell biochemistry and immunohistochemistry as well, and also everything from giving us reasons to hit the gym, to live longer, to
do not eat too much, and especially not sugar.
What to do when you have insignificant data, the trend, very nice, and everything in between, as well as the ethics of stem cells. So let's thank our speaker once again. So the speaker will stick around for a few minutes here. If anybody wants to talk to him one-on-one, you're welcome to do so.